work on using neuropixels for in primates. I think Gabriel himself works on decision making. Is that right?
Half right.
Half right. Okay, good. So yeah, okay, good. Wait, I'll remove that. And we're all set. Good. Thank you very much, Gabriel, for coming. Over to you.
Can you guys hear me? Yeah, great. Yeah, thanks, Matthias, and all the other organizers for putting this together and having me. Yeah, I'm very excited to talk to you all about what I really consider a new era for primate neuroscience using these newly developed neuropixels NHP probes. I'm currently a postdoc in Mehrdad Jazaeri's lab at MIT. My role in this project was mainly through my PhD work in Mike Shadlin's lab at Columbia. So I'll start off with a little bit of history. There we go. So I assume most of you know about the really foundational work of Hubel and Wiesel discovering receptive fields and orientation selectivity first in cat V1 and how this sort of ushered in a new era of neuroscience in the 1950s. But what's a bit less appreciated is that this discovery was preceded by a couple of years by the invention of the tungsten microelectrode by David Hubel, which he published in Science. And these electrodes sort of gave scientists a way of doing new experiments that people weren't thinking about before, and sort of set up the whole field of systems neuroscience for several decades following that. And it's kind of amazing to think how much we have learned from recording, one neuron at a time with these single electrodes. But at the same time, they're also extremely constraining. So especially for work in monkeys where the technology has lagged a bit. So single electrode recordings, constrain primate neuroscience for decades. Specifically, they require huge time commitments. These projects usually involve doing months to years of recordings almost every day. It's a real drag. Because you're only getting one neuron at a time, you're heavily relying on trial averaged activity. Experiments where you want to do circuit dissection is extremely difficult. You often require these heroic sort of antidromic stimulation experiments where you get something like 5 neurons in 6 months. You get this bias sampling of neurons that Andrew discussed a bit. And you have stability issues with single neuron. The neuron sort of travels with the brain and you lose it. And so with all these constraints, you sort of ask yourself, why on earth would anyone do monkey neuroscience? I mean, it's just so difficult. And in addition to that, the tools we have sort of set the parameters for the types of ideas we get, right? So the hypotheses, the experiments we plan are all in the context of the tools that we have. And our ability to be creative depends on the tools that we have. And so what we think is going to really help or maybe solve many of these constraints are these new NeuroPixels NHP probes, which I'll tell you about today. And I just want to start with saying that this was very much a huge team effort led by Eric Troutman, who was initially invited. I'm here sort of in his place. So big thanks to Eric. And this was a big team effort, a collaboration between the Howard Hughes Medical Institute and IMEC. And my role in this was one of the main testers of the probes in monkeys, trying to figure out how to get these into the macaque brain and really get them to work and analyzing the data and figuring all that out. So today I'll tell you first just the general specs of these probes and how they perform in the macaque brain. I'll spend most of my time telling you about the new capabilities that these probes enable. Mainly, I mean, one of the obvious ones, you just get more data more easily, which is a huge benefit. But what I'm most excited about is just the huge expansion in the space of possible experiments that these probes allow. Lets you do experiments that you really just would never even consider before. Which I think is going to be the main impact. on the monkey neuroscience field. And then finally, I'll talk a bit about resources that we've developed to support widespread adoption, right? Any new technology is sort of as good as it is to, or its impact is sort of limited by how easy it is to use. So we've done a lot of work to try to make this as plug and play and easy to adopt as possible. So the main version of the probes I'll be talking about are these 45 millimeter versions. So they're very much like NeuroPixels 1.0, like Andrew talked about. They're just longer. There's two versions. There's this 45 millimeter version, which is 125 microns wide. So it's, I think, over double the mouse versions. So they're quite a bit more sturdy than the mouse versions, which helps with things like penetrating the thick dura of the macaque. And these probes in total have about 4,400 channels across the full 45 millimeter shank. There's also a 25 millimeter long version, which is a bit thinner, 60 microns thick. And these have 2,500 channels across the whole shank. And just like the 1.0 probes, there's programmable site selection where you can record from 384 channels simultaneously. These probes are currently available. Here's the pricing data. Apologies for not converting this to pounds before my talk. I thought about it and then didn't do it. And the full system cost for getting all of the National Instruments equipment and the probes and head stages comes out to about 10K right now, but that should be decreasing soon with hopefully the one box will be coming out soon that IMEC and HHMI are developing. And the last thing I'll say is that these probes can be used indefinitely. They really last as long as you'll take care of them for. My PhD work, I used one single probe for every single experiment I did. So like I mentioned, compared to the mouse probes, these are simply longer. And here's just a very clear depiction of why that's necessary for doing monkey recordings. So this is a picture of a rhesus macaque brain sort of at scale with the probes. And it is actually possible to get a mouse 1.0 probe into the macaque brain, but man, is it hard. It's just not worth it. And so these long probes make recording from most of the macaque brain much easier, and it enables recording from almost all deep structures in the macaque brain as well. And just like the mouse probes, they have different banks of channels that you can record from along the whole shank, consisting of 384 electrodes each. And I think one sort of important difference in the monkey brain with the mouse brain is just because the monkey brain is so much bigger, for most of your recordings, an entire bank is within the brain area of interest. So you really get almost all 384 channels in one brain area. So it really helps with the yield. So also highlighting things that Andrew discussed a bit is that the different banks you can sort of select different configurations between them. I think the most common configuration is just recording from the densest, deepest bank on the left. But you can get creative. You can record from a single column across two banks to increase your recordings from 7 to 7.5 millimeters, and go up to 15 millimeters. Or on the right, you can record from two separate banks, say one at the surface of the brain and one deeper in the brain. That helps with multi-brain area recordings. And this was very sort of intuitive and easy to select while you're doing an experiment. You could even put the electrode in the brain, search across the different banks to see which has the best activity, and record from that one. I'll also mention that the probes come pre-sharpened now, so IMEC will sharpen them before they ship them out. And so these probes can penetrate the dura. People in our lab and other labs do this regularly with little issue. So here's some raw data collected by Eric Troutman and Dan O'Shea when they're in Krishna Shenoy's lab. And here I'm just showing you the voltage traces across all 384 channels for a span of about 20 milliseconds. And the type of data that we get in this raw data is just simply beautiful. I've zoomed in on two example units. And so you'll notice that you get multiple channels capturing the same action potential. And you might think that this is maybe a gross oversampling of units. across this, but it turns out it's extremely helpful for spike sorting, as I think many people in here appreciate, with drift correction. And also it allows you to sort of more precisely locate the location of the waveform on the probe. And so here I'm just showing you 2 example units now that were pretty close to each other. And what I want you to appreciate is that there are a few channels here where if you didn't have this same density, that it'd be extremely hard to tell them apart using spike sorting, like this location. Oh, that's laggy. Okay. Like the waveform on the bottom, but by having the high density, there's enough channels where the waveforms are separated, making it simple to spike sort. So across a session, we get about 50 to 400 neurons, single well-isolated units per session. Here's 2 examples recorded from monkey primary motor cortex, again, recorded by Dan O'Shea in this case. And that's about an order of magnitude more than what we were getting with our previous technology in the monkey. And sort of drift and motion artifacts are often a problem with these types of recordings. And at least in the monkey, we've been able to develop protocols that give us super stable recordings across several hours. So on the left, I'm just showing you the cluster depth as a function of time over about an 1.5-hour recording session. So it's super stable. On the right is the estimated probe drift, and yeah, we can get less than 25 microns of drift within several hour session. So those are the probes. I'd love to tell you now about the sort of game-changing science that we can do with these, and I'll sort of give you a few sort of short vignettes about different labs in our cohort that have used these to just do what I think are simply amazing experiments that are actually not all that hard now. So the first is using the ability to select multiple banks to record from more than one brain area at the same time using a single probe. So if you plan your trajectory carefully, you can, for example, have a single probe go through motor cortex and the globus pallidus of the basal ganglia. This was a experiment conducted by Andrew Zimnick and Eric in Mark Turchin's lab. And you can go even further, and Shude Zhu and Rubeng Jia in Tiran Moore's lab have used a single probe in the STS of a monkey to record from 3 visual areas in a single session. In this case, they're switching between 4 banks. So they'll record from one bank in one area for a while, switch to another one, switch to another one. And this is the type of data that you get when you do that experiment. Here I've flipped the probe sort of on its side so I can fit it onto the slide. And each of these different colored lines is a different waveform that they get across the different banks. And here they're able to get over 1,500 neurons across 3 brain areas in a single session of recording. This is something that would take several years to do with previous technology, maybe not even possible. And not only that, but you can look at the receptive field structure across this whole probe and just see this amazing structure across the probes. You can see the transition from V4 to MT. V4 has these sort of less well-defined receptive fields on the top, where MT has these clear, isolated, spatially isolated receptive fields. Within MT, you can see the sort of drift of the receptive field as you go deeper into the STS, and so on. And I mean, there's no real, not a huge insight here, but just trying to give you an idea of just the amazing types of data that you can get from this, and just the new opportunities that having this much data this quickly allows for. Another example comes from Doris Sau's lab, where Frank, Giannis, and Daniel did these recordings where they do these amazing experiments where they have a monkey in an fMRI scanner while the electrode is in the brain, and they're able to precisely target face patches of the monkey brain. In this case, they record from face patch ML and get up to 250 units, most of them having face selectivity. And then they can put a second probe in and record simultaneously from face patch AF at the same time. And I just want to point out that face patch AF is over 3 centimeters deep into the brain, and they reach this no problem. And they can, record from these many phase-selective neurons from 2 different brain areas simultaneously. And again, the types of data that they get is just like almost hard to believe from a monkey neurophysiology standpoint. So here I'm showing you a raster. This might look like your sort of typical standard raster that you see in many neuroscience papers, but this is actually a single trial that you're looking at over several hundred neurons from 2 different brain areas. And so this is sort of what vision scientists have always wanted to be able to see, and we've always inferred by using trial average data, but it's now accessible to us, right? So we now have single trial resolution available in the monkey brain. And single trial resolution is obviously fantastic for vision experiments, but I'd argue it's even more important for experiments on decision-making and cognition, which is what my work was on. Behaviors like decision-making. So imagine a monkey is doing a random dot motion discrimination task where a monkey is fixating. It views a bunch of randomly moving dots on the screen. And whenever ready, the monkey has to indicate its decision with an eye movement, in this case to the right. And we know that these types of decisions are well characterized by a bounded evidence accumulation process where information from the sensory stimulus is sampled sequentially and integrated over time until a threshold for making a right or left choice is exceeded. In this case, here's an example of a rightward choice. And critically, this sort of integration process, this decision variable that you're seeing here, maybe, is different on every single trial, right? We generally average over many trials, but the theory is that the trajectory of your decision is unique on each trial. Now, for decades now, labs like Mike Shadlin's lab has done recordings in the lateral interparietal area, or LIP, and shown that these neurons have characteristics that match this evidence accumulation process. So here I'm showing you the average over many neurons over many trials aligned to the onset of the motion stimulus. And activity in LIP sort of ramps up or down depending on the strength and direction of the motion. But really what we hypothesize these traces to represent, and I'm just going to highlight one of these averages, is that, well, this might be what the average over many trials looks like. What we hypothesize is that this average is comprised of single drift diffusion traces. So this is what theory sort of predicts it should look like, but because we haven't had single trial resolution, we haven't been able to directly observe this drift diffusion process. So now with NeuroPixels probes, we're able to record from enough neurons to get this sort of single trial population level data that we need. This is work I did in collaboration with Natalie Steineman in Mike Shanlin's lab. And just to give you an idea, like I mentioned, these probes, right, you have 384 active sites. We would get 50 to 300 neurons per session in LIP. And LIP has spatially receptive fields like other visual areas, but it's not highly retinotopically organized anatomically. So when you put this probe in, you get response fields or receptive fields all over the visual scene. And historically, we've recorded from or searched for the receptive fields that overlap one of the choice targets that the monkey uses to indicate its decision. And so with previous technology, you would do this and you'd get maybe 1 or 2 neurons or 0 neurons that had a receptive field in a particular location. And here we get something like 5 to 40 neuron or target in or TN neurons from these NeuroPixels probes. And so here's an example session from one of these recordings where we got 17 of these target in neurons. I'm showing here the trial average data just like before, so it looks very similar to what we've seen in the past. But now we can open sort of, right, and again, just highlighting one average, we hypothesized the single trials to look like simulated drift diffusion paths. So now we can open up the box and ask, well, what's actually happening on a single trial? And so here's, if I just take the average of these 17 neurons in a single trial and plot it, this is what one trial looks like. Here's what all of the trials look like. And I hope you appreciate just the visually striking similarity between the predicted diffusion and what we observe in the single trial data. And similarly, if we align the data to the saccade, the time that the monkey is committing to a decision, and again, we highlight just one of these averages and ask, well, what do the single trials that comprise this average look like? They look something like this. And just like people have observed in trial averages, we see this sort of point of minimum variance in the single trials, which has historically been interpreted as some downstream area applying a threshold to the LIP signal, which then leads to commitment. So we wanted to test this threshold idea and ask, well, can we record from LIP's downstream target and test this idea? So we then added a 32-channel linear probe into the superior colliculus, one of LIP's main targets. And the superior colliculus, unlike LIP, is highly retinotopically organized. When you put a probe there, you get dozens of neurons that span or that cover the choice target. And so now we can do this experiment where we can have simultaneous recordings in LIP and the superior colliculus with overlapping response fields. And this was an experiment that simply wasn't possible before the NeuroPixels. And when we do this, we can look at the trial average activity again. It looks qualitatively similar to LIP, except we get this saccadic burst of activity, which has been known for decades in the colliculus. But again, we can peer into the single trial dynamics. And what we see here is, again, this large saccadic burst of activity. But what we see in the single trial data, and which wasn't apparent in the trial average data, are these non-saccadic bursts of activity that sort of occur at random times throughout the trial. And because they're at random times and the averages, they sort of get canceled out. And the main takeaway I want you to take from this is that we see similar trial average activity, but distinct single trial dynamics. So it sort of begs the question of the types of things that we might be missing by focusing so much on trial average activity and just the insights that looking at single trial dynamics might afford. So we wanted to take this one step further, right? This led to the hypothesis that SC implements the decision threshold, where an LIP represents the accumulation of evidence. Well, can we test this causally? And again, the NeuroPixels let us do an experiment that wouldn't have been feasible beforehand. So now we're going to inject mucimol into the superior colliculus and inactivate it. And at the same time, we're going to record an LIP with a NeuroPixels probe. When you inactivate SC, you get what we're going to call an inactivated region. So there's a spatially localized part of the visual field that eye movements towards are affected. That's the orange region. And with the NeuroPixels probe, we can get dozens of LIP neurons that have a response field within the inactivated region and that overlap the choice target that the monkey uses. Now, in this experiment, the hypothesis we put forth is that if the colliculus is terminating the decision, implementing the decision threshold, then inactivating it should raise that threshold, right? And more evidence would need to be accumulated to make the same choice. And in LIP, what we would then predict is if we align activity to the time of the saccade, the time the monkey's committing to a decision, this is what it looks like before we inactivate SC. And this is what it looks like after SC inactivation. And it's as if the threshold that's being applied to LIP was being raised by SC inactivation. So consistent with the idea that SC is implementing a decision threshold. And again, the main point here is that the large yield of NeuroPixels enable experiments that were just simply not previously feasible. And it's not just getting more data more easily, it's that we did an experiment that we would never even consider beforehand because of the yield that we have. So one other sort of new feasible approach that I want to highlight is the ability to stimulate and record at the same time. Dan O'Shea in Krishna Shenoy's lab did these experiments where he had a NeuroPixels probe in monkey motor cortex, and at the same time had a stimulating electrode in the same region and was stimulating while recording while the monkey was doing a motor task. And when you stimulate and record at the same time, sort of unsurprisingly, you get these giant electrical artifacts from the microstimulation that completely drowns out all of your spikes. But because the NeuroPixels have this extremely high density, you can develop relatively simple algorithms to cancel out the stimulation and recover what the spiking activity looks like underneath. And so this is with an electrode in the same area as M1. And I encourage you to look at, Dan has a new, relatively new paper with Lisa Dunker that I recommend you checking out. It's fascinating. And I've used the similar approach in my postdoc work with Mehrdad Jazayeri to ask how the cerebellum interacts with its downstream target in the thalamus, specifically the dentate nucleus. And so currently I'm doing experiments where I have a neuropixels probe in the ventrolateral thalamus, and I have a stimulating electrode in the dentate nucleus at the same time. And we'd like to identify the subregion of thalamus that's receiving input from the dentate nucleus. And historically, this would be an incredibly difficult experiment to identify this region, with one neuron at a time, or a few dozen at a time. But with neuropixels, we find readily many neurons that are driven by stimulation. So here's an example neuron in the thalamus during or not during stimulation of the dentate nucleus. So very clear activity being driven. And we can sort of move the neuropixels probe to different locations in the AP axis in this case, and just see a very sort of parametric change in the proportion of connected neurons. And so we can be reasonably confident when we're in a region of thalamus that has large target from the dentate nucleus versus lower magnitude target. And so again, just the type of like circuit dissection experiments that we've always wanted to do in the monkey that, it's sort of simple to do, or much easier to do in the mouse, it is now more accessible in the monkey, which I think is going to be a real game changer. So just to end my talk, I'd like to, again, discuss a bit about the resources we've developed to support widespread adoption. I think really the biggest thing is that these neuropixels probes are sort of plug and play with the whole rodent system, right? There's really no difference other than they're longer. So by adopting these probes in the monkey, you'd sort of be joining this already thriving and supportive community that's already using neuropixels. There's a really fantastic course available on YouTube from UCL that I've found incredibly helpful. There's a Slack channel, which is very active and incredibly helpful. And so I think like it's hard to overappreciate just how beneficial this community can be for people adopting a new technology. Second is that the cohort who developed or tested these probes, we've designed sort of custom hardware to interface the probes with the types of microdrives that people are using already. So we have open source designs for Thomas recording microdrives, for Nan and Narashige. And so we're hoping that for most use cases, people can simply download one of these designs, get it manufactured, or even 3D print it, and just swap out whatever probe they were using and attach the NeuroPixels probe instead. So we're hoping that that'll be as straightforward as possible for simple use cases when you have a single probe in one chamber. For more complicated setups where you might want multiple probes or multiple angles, we've also open sourced our designs for these. Eric has been able to record from 8 neuropixels in a single chamber. That's that image on our right. So if you're feeling particularly ambitious or crazy, you can try downloading that and figuring that out. There's designs from Janis Hess and Doris Sauslab in the middle for getting extremely deep recordings, as deep as 42 millimeters into the brain. So almost the entire probe is in the brain. And he's figured out a solution for that. And I've figured out a similar solution on the left for that. And all these designs are either currently or will be available in this GitHub link below. It's also available in the preprint that I linked earlier, which I'll show again later. And if you have any questions, feel free to email any of the authors or link or reach out to the monkey specific channel in the NeuroPixels Slack, which is also active. And then finally, a project that I've been working on with a PhD student in the lab, Jack Gable. We're working on what we call Monkey Drive, which is this a new open source drive that we specifically designed for using NeuroPixels. We're just trying to make this as simple as possible for people who want to use this technology in monkeys. It comes with the drive itself. We've designed free intuitive software. We'll have hardware available. And it's very much still in development stages, but this should be ultra low cost. I think we're talking about an order of magnitude cheaper for what the current options are. Even for up to 8 drives is going to be less than $5,000 or $10,000 for the whole system. So with that, I'll summarize. I told you about these new NeuroPixels probes for monkeys that enable deep, large-scale recordings in the monkey. They enable these sort of amazing new experiments that we could have never done before, which is going to change primate neuroscience. And we've hopefully made getting started as easy as possible. And again, I'll just advertise I'll be at the session tomorrow. So if you want some practical information on how do you actually get these things going, please talk to me tomorrow or at any other time during the next few days. And I'll just sort of revert back to the David Hubel's invention of the tungsten microelectrode, which ushered in basically systems neuroscience, or invented systems neuroscience and set it up for decades. And I really think that NeuroPixels as a whole is doing the same thing, maybe already has, and especially for the monkey neurophysiology research. So with that, I'll again just thank this really large group effort. I was a tiny part in this huge team. I was very thrilled to be able to tell you about it all today. Again, a lot of the credit goes to Eric Troutman, who really drove this forward, who, like Matthias mentioned, might be on Zoom at that session tomorrow. Of course, the funding sources and all my other co-authors on this. I'm happy to take questions. Thank you. Mm-hmm. Mm-hmm. Yeah, so in that case, it's probably possible to find a single trajectory that would do it. One thing I'll say is that because the colliculus is such a highly organized retopic map, you'd really like to approach the colliculus in a way that's like normal to that map so that all the neurons have the same receptive field. And I don't think you could find a trajectory through LIP that would let you do that. In our case, it was just simpler to split it into two.
I don't know much about monkeys, so maybe very naive, but how do you actually target your location you use in your lab?
Yeah, so different labs have different solutions. I think one of the more popular solutions is you first do a structural MRI, and you use that to plan your surgery for where you're going to place your chronic implanted chamber. And then after you do that implant, if you're using one of these PEEK plastic chambers, you can do a second MRI, which has the recording grid you have in there to really visualize the trajectory you're using. It's especially important for deep areas, right? When you're going 40 millimeters deep, a few degrees of error in angle, you'll be somewhere in a completely different brain region.
Right? No more questions? Great. Thank you again, Gabriel, for coming.